Hello, I'm Jochen Sieg. I'm from the Center for Bioinformatics at the University of Hamburg. I'm part of the group for computational molecular design of Professor Matthias Raray. And first of all, I want to thank the organizers of the ISMB conference that make it possible that we can present our work here. And now I will talk about data-driven analysis of single-point mutations through rapid scan of 3D microenvironments. As probably all of you know, single mutations can have a large effect on the overall protein. For example, its stability, binding to other proteins, or ligands. In structural bioinformatics, mutations are often modeled through changes to the local microenvironment, the direct neighborhood in the protein structure, instead of considering the whole protein complex. Another common pattern is an asymmetric use of experimental structures. This means usually there is not a protein structure for both the wild type and the mutant, but only for the wild type. To address this issue, we developed a new tool for mining experimental mutant structures based on 3D microenvironment database search. This new tool is based on the existing Sienna and Ascona technology, which was also developed in our group and was originally developed for searching binding site conformations in large protein structure databases. We modified and extended this approach to search for mutation structure pairs based on 3D microenvironments. In the workflow of this new tool, we start with a protein structure representing the wild type. For each residue, we use the local microenvironment separately to search for similar environments in the database that have a different residue at the position of the query residue. The result of the workflow are structure ensembles for each residue of the input. And these structure ensembles are exemplifying the local effect of a mutation. And now to better illustrate how the algorithm works, let's walk through one example of searching with a single query microenvironment. So we start with a single residue for which we want to find mutant structures. So we have query structure, we have our single query residue, which is marked here in red. And the first thing we do is we compute the local microenvironment of this red residue by collecting all the residues in a distance of 6.5 angstrom to our query residue. This is the microenvironment. We then generate connected sequence fragments from these residues by connecting and extending shorter sequence fragments. Each sequence fragment is then used for a k-mer-based database lookup. And since we want to find mutants, we discard all the k-mers that are overlapping the query residue and generate new k-mers with the other 19 amino acids at this position. The result of the k-mer step is a list of database IDs representing candidate structures from the database. And each candidate structure is then evaluated in detail with the Ascona tool. The Ascona tool first aligns the query sequence fragments to the chains of one candidate to generate an initial sequence-based matching of query fragments with candidate fragments. This initial matching is then optimized geometrically by the rotational distance of matched fragments using quaternions, and also by the deviation of the structural arrangement of the current matching with the query by comparing all pairwise distances of fragments in the query with the respective pairwise distances of their matched candidate fragments. And in this way, we can retrieve microenvironments that are identical in sequence except for the position of the query residue for which we want to find a mutant. And these microenvironments should also have a similar structural arrangement. Example of how such a result looks like is illustrated here on the slide on the right. We have here the blue structure, which is a result from the database search. It's superposed based on the microenvironment on the query structure. And as we can see here, the local microenvironment is highly similar. And what we can also see from this picture here is that also the whole structure is globally very similar. While this is probably a frequent case of structures that we find, we can also find structures that are more different globally, and we can find them as long as their local microenvironment is similar. Now that I have illustrated how the algorithm works, let's look at two more examples what this algorithm can find in a database search. In the first example here, I have a monomeric stephin B, a protein that usually acts as a cysteine protease inhibitor. We use the environment of protein 79 here as a query to our tool. And from database search using the microenvironment here, we get one result that is, I think, quite interesting. We see here the superposed structures. Here is the query residue, the protein, which is mutated to serine here. And what we also see is the loop, which is also the query structure, and that is superposed here based on the microenvironment from the database structure. And what is interesting here is that while these microenvironments are identical in sequence, except for the mutation, of course, the orientation of the side chains in the local backbone is switched in this case. And that we can find such a case is likely because we focus on high sequence identity in the environment, while we at the same time have a not so strict structural filter. And therefore, we can also allow to find more structural deviating results. And the second example I want to show here is an example of a protein-protein interface. Here you see the trypsin complexed with trypsin inhibitor. So the trypsin is on the left and inhibitor is on the right. And what I want to illustrate here is that when we use a residue at the protein-protein interface, the local microenvironment will contain fragments from both chains. And this means that we can specifically search for mutations in protein-protein interfaces. What is interesting in this particular case, when we search with lysine 15 of the inhibitor, we find in the database that this residue is experimentally well explored with mutations, because we can find 12 other residues from the PDB, which we then can superpose, and we can look at the structure ensemble. And for example, we can compare how the different residues interact with their respective microenvironments. And now, after we have looked at different structure examples, now let's switch to looking at evaluation on large data sets. For the evaluation of our tool, we use 5 mutation datasets. Specifically, we use the well-known proteome dataset. We also use the ThermoMuteDB, which extends the proteome dataset and was released this year. We use the Platinum dataset, a dataset that contains mutations on protein ligand complexes. And we use the proteome dataset, which was also released this year and extends the proteome dataset. Finally, we use the SCAMP2 dataset, which contains mutations on protein-protein interactions. While all of these datasets contain mutations from different sources, we are here only interested in unique single mutations with known wild-type structures. These are shown here in the second row. So for example, for the proteome, there are roughly 4,000 different single mutations with known wild-type protein structure. And for our first experiment, we are also only interested in mutations for which both a known wild-type and mutant structure is available. This is the table, the third row. And as you can see, only the first 3 data sets here have these annotations for their mutations. And what we want to do now is we want to know how many known wild-type mutant structure pairs we can retrieve with a tool. And what we did first was a baseline experiment using only the Ascona tool. with the protein structure pair of the wild type and the mutant as input. This means we do not yet perform the database search, but we only want to evaluate whether we can identify the specified mutation in the dataset, the only thing that we want to evaluate is whether we can identify the specified mutation in the mutant structure given the wild type structure using the tool. And as you can see here in this first experiment, the mean retrieval rate of all the data sets is 86%, which I think is already quite good. However, we were interested in the cases that we failed, why we failed in these cases. So what I did is I took the structure pairs, loaded both structures into a viewer, superimposed them globally, and manually looked at the position where the mutation should be. And interestingly, in many cases, there was the same residue at a specified position. And after removing these cases and rerun the experiment, we get a mean retrieval rate of 98%. And then next, of course, we wanted to know how well the overall workflow, including the database search, performs. And here we got a mean retrieval rate of 90%, which I think is already quite good. This shows that we are this also shows that we are losing 8% due to the database heuristic when we compare this experiment to the baseline experiment. And this illustrates that there's a point for improvement. And in this case, I think this can be improved. However, 90% is still a good performance and probably already sufficient for many applications. And one such application I want to show now. We want to use our tool to annotate the mutations with known wild-type structures in the 5 mutation datasets with mutant structures from the PDB, such that in the end, they have both a protein structure for the wild type and for the mutant. The results of this experiment are shown here for the protein dataset, for example. You can see that we can, for the protein, annotate 726 mutations with a protein structure for the mutant out of roughly 4,000 mutations. This is an increase of this coverage, this annotation, from 7% to 18%. And when we look at all the 5 data sets, we see that we can improve the annotation of this mutant structure coverage by at least 2.4-fold here over all the data sets. And I think this is already a quite interesting result here, because this increases the number of training samples that can be used, for example, for building predictors for stability changes upon mutation. But what I also want to show here is that in addition to just annotating the mutations that are present in the mutation dataset, we also find many different wild-type mutant structure pairs in the PDB, which are not necessarily included in the mutation datasets. And just to give a short impression of what data is available in the PDB, when we use the wild-type protein structures from the protein for search with our database search tool, we find all in all 4,519 different mutation structure pairs. And for other data sets, we find even more than 10,000. So it seems that there is still a lot of wild-type mutant structure pairs in the PDB available, and that might be very useful for our applications. And with this, I want to come to the discussion and conclusion of my talk. We have here proposed a new tool for analyzing single mutations with experimental structure pairs based on highly similar microenvironments. We have also seen a first application for this new tool to annotate mutant structures to thermodynamic mutation data. And however, I think that there are many other applications imaginable where such structure pairs would be valuable, for example, in structure modeling, sidechain conformation prediction, or even in other areas like research on resistance mechanisms of relevant drug targets. In every scenario where mutations are important and where experimental structures of wild type and mutant can have a benefit. Our tool will be made available as command line tool and at our web server, the Proteins Plus. And finally, all in all, I hope that we can now, with our tool, more easily provide experimental structures to studies currently lacking in mutant structure. And with that, I want to thank my group in Hamburg, specifically Professor Matthias Raray and Stefan Dietz. And of course, I want to thank the BMBF for supporting this project. And if you're interested in our software, you can check out those links. And finally, I also want to thank you for listening.